So let's go now to the classification of the chromosomal aberrations that we have described before. So first of all, chromosome type aberrations, so the aberrations that involve both chromosomes at the same point, essentially only occur when the cell is exposed in G0, G1. So when there is only one chromatid per chromosome, and then the aberration will be duplicated in S phase. On the other hand, chromatid type aberrations generally occur in late S and G2. So when you have two chromatids, so one can be affected and the other no. There can be interchanges if there is exchange of genetic material between chromosomes, or intrachanges if the exchange of genetic material is within the same chromosome. They can be symmetrical and asymmetrical, simple type and complex type, we will see this. And then they can also be complete or incomplete forms. In the incomplete forms, essentially, there is an unrejoined piece. We have seen in the rejoining of DNA double-strand breaks, repair proficient cells generally do not show any residual unrejoined break. But if there is a residual unrejoined breaks, this would be an incomplete exchange. So let's start now to see the most common chromosomal aberration. One of the most famous is the dicentric. The dicentric is classified as an asymmetrical interchange. Why is it an asymmetrical interchange? It's an interchange because it occurs between two different chromosomes, and is asymmetrical because the result is two different structures. In the classical theory of radiation action, let's say what is really called the classical theory of radiation action was developed by Douglas Lee in 1946. The idea is that radiation breaks the chromosomes. Now we can translate it in a more modern wording. We say radiation-induced DNA double-strand breaks. And during the repair of these double-strand breaks, maybe because of alternative non-homologous joining, you can make mistakes. For example, if you break two chromosomes, we are talking about in G1 here, okay, chromosome type. So every chromosome is only one chromatid. If you break the blue and the yellow, instead of having proper rejoining, you can make a mistake and you can misrejoin the blue with the yellow. When you go through the S phase, you duplicate this dicentric. You see, this is a PCC. So the PCC here shows exactly this situation. You see the orange and the blue, and there is also a centromere probe to see the centromeres here. You see orange and blue dicentric chromosome, and you see here the acentric fragment, and you see here the other chromosome, the normal chromosome here with his centromere. Eventually will become a dicentric fragment with an dicentric chromosome with an acentric fragment, and this is how it looks like in ENFISH. You see, this is a dicentric between chromosome 2 and chromosome 9. You see here the chromosome 9 with the centromere fused with the chromosome 2 with the other centromere. Now, you can then have asymmetrical again, but not interchanges, but intrachanges. You have two types of asymmetrical intrachanges. One is interarm, and in this case is called a ring. What happens in a ring? You show it here. You break the chromosome here and here, and then you rejoin the two pieces along with the centromere. So you generate a ring. This is in RX fish. You see, this is an RX fish image, very nice, because you see, this is the chromosome number 3, I believe. And is broken, you see, this broken here is broken here and here. These two broken pieces rejoin together to form this acentric fragment. And then this piece rejoins with this piece to form a ring. Rings are sometimes found, you see, also in amniocentesis. This is a G-bending image with the karyotype from an amniocentesis, so from a baby, where there is a ring in chromosome 14 that can be actually mapped very carefully with G-bending to the band P11.2 Q Q32. So P11.2 means P-arm band 11 sub-band 2, Q32 is the band in the Q-arm. Dicentrics are asymmetrical interchanges. What about symmetrical interchanges? Symmetrical interchanges are called translocations. And of course are exactly the same as dicentrics. You break one chromosome here, you break one chromosome here, but now the rejoining is not joining together the two pieces with the centromeres. It's just exchanging the other two pieces without the centromeres. Why they are very famous? They are very famous because translocations are very often found in cancer, very often. One of the most famous translocation, which is somehow represented also here, is the so-called Philadelphia chromosome translocation, which is responsible for the chronic myeloid leukemia. In chronic myeloid leukemia, you have a translocation between chromosome 9 and chromosome 22. The derived chromosome, you see this chromosome here is called chromosome Philadelphia and is responsible for is actually responsible for leukemia, because what happens in this translocation is that you move a proto-oncogene, the ABL oncogene, the CABL, cellular, because there is also a viral version, the VABL. This is the CABL. With this translocation, you move the ABL chromosome gene downstream of BCR, which is a strong promoter. So with this translocation, you promote ABL, which is an oncogene, so promote the growth of the hematopoietic stem cells, and you have leukemia. You see translocation very well, of course, with ENFISH. ENFISH is the best way to see them. In this slide here, you see a very beautiful translocation between chromosome 1 and chromosome 4. You see reciprocal translocation. These are complete, not incomplete form. So you see the two pieces are perfectly rejoined. 1, 4, and you see a little piece of 1 here, and a piece of 4 here. And then you have a 7Y. You see the Y as a very strong and bright Q arm. And you see a little piece of 7 down here. So there are two translocations induced by radiation, actually, in this cell. Now, we have seen before inter-arm intrachanges, but we can also have symmetrical inter-arm interchanges, not necessarily asymmetrical. You remember the asymmetrical inter-arm interchange was a ring, but you can have a symmetrical inter-arm interchange, which is called pericentric inversion. You see, in the pericentric inversion, essentially you have again a break in the P-arm and in the Q-arm, just like for the ring, but now you swap the part with the centromere. So now you will have is symmetrical because the result is exactly the same. You see very nicely if you use arm-specific probes, you see it very nicely because you see the normal chromosome will be green, P-arm is green, Q-arm is red. If you have a pericentric inversion, you will have green, red, of course, because you have to switch the chromosome here, you break here, and so you change the order. So arm-specific is a very nice way to see the pericentric inversions. You can also see in RX fish, for example, here you see this is a chromosome 7, and this is a chromosome 7 after a pericentric inversion. Again, you see the pericentric inversion, this is a band here that now you see the change. Paracentric inversion instead are intra-arm symmetrical interchanges. So now it's a symmetrical interchange, but it's not inter-arm, it's intra-arm. So now you have the two breaks are both induced in the same arm, in this case in the P-arm, and then there is a swap. To see them, the best technique is, of course, is the G-bending, because the arm-specific fish, you will not be able to see them, because, of course, it's the same color. But if you have M-bend, you see here, you see an inversion in the order of the bands that tells you that there was a paracentric inversion. And then you have complex type exchanges. By definition, a complex type exchange is an exchange that involves more, at least, if you want, 3 breaks in at least 2 chromosomes. This is a definition by John Savage, who was an English cytogenetist. They can be non-reciprocal exchanges and insertions, include intra and interchange events, and they have various degrees of complexity. This is just to give you an example. This is a normal chromosome in N band, and this is now a large insertion of a piece of another chromosome. I don't know which one, because of course this dot is blue, was inserted between bands 6 and 7. You see that is a complex exchange, because how many breaks you have? You have 1 break here, and you have 2 breaks in the original chromosome. So you have 3 breaks in 2 chromosomes. So it is actually a complex exchange. As well as this one, this quadricentric, this monster chromosome, if you think about, this one comes from 4 breaks in 4 chromosomes, can only come from 4 breaks in 4 chromosomes, so it's a complex exchange. And this one, which is a dicentric plus an insertion, you see that if you use Giemsa staining, you would just classify this one as a dicentric, but it's much more complex. It's a dicentric plus an insertion. Then you have the deletions. There are 2 types of deletions. One are the simple terminal deletions. Terminal deletions are incomplete exchanges, if you want. So you break 1 chromosome. This chromosome is not rejoining with any other chromosome. And you have a situation like that, that you see the chromosome and you don't see the telomere signal anymore. It's like lost. You see, because clearly you have lost this piece of the chromosome. And you can also see it here. But you can have, this very frequent, interstitial deletion, where you actually lose a piece of chromosome from the internal part of the chromosome. And actually, this interstitial deletion is not shown here, I'm sorry, but they are actually intra-arm asymmetrical interchanges, because normally this piece, you see this band number 5 that you see here, rejoins and forms a little acentric ring. So this is, in principle, is like an intra-arm asymmetrical interchange, also called interstitial deletion. And finally, you have the chromatin type aberrations. If you go to SG2, in this case, you can have damage only in 1 chromatid, not in both chromatids, and you can have this kind of different rejoining or breaks involving the single chromatids. The picture is very complex at this point with chromatid type exchanges. You see, you can have, some of them looks like gaps, breaks, you see the broken chromatids, exchanges of different types, and also complex type exchanges, but chromatid type, not chromosome type. You see another example here. So and you can see them, of course, in different techniques. This is with ENFISH, a complex type exchange in ENFISH in a mouse cell line. And I can tell you it's a mouse because the mice, unlike humans, all the chromosomes in a mouse are acrocentrics. There are no metacentric chromosomes. And you can see here a complex chromatid type exchanges involving 3 different chromosomes. You see 14, 17, and 1. So, but why we are so interested in the chromosomal aberration? Why is so easy? I mean, historically, you know, the study of chromosomal aberrations comes before the studying of DNA damage and repair, because it's much easier. It's something that you see at the microscope. But now we understand that they come from DNA damage and repair. But the reason why chromosomal aberrations are so important is that there are early radiation events then associated to late cellular consequences. both cell killing, so they are important for radiotherapy, for example, and transformation, so they are important for radiation protection. Generally, let's say the first statement to do is that chromosomal aberrations are not good for humans, of course. The presence of chromosomal aberrations in our body, especially if it is constitutional type of aberration, is associated to disease, in some cases, very serious disease. The most famous by far is certainly the Down syndrome. The Down syndrome, which is, as you know, 47 plus 21 karyotype. There are a few cases where it's not really a numerical plus 21, but it's a translocation 1421 or 2122. The frequency is pretty high because it depends very much on the age. It's fairly rare, less than 1 in 1500 for young women. But when the maternal age is higher than 40, becomes higher than 1 in 100. So it starts to become a high risk of Down syndrome. And you know also the phenotype, of course, you know, I'm sure you know many people with Down syndrome because it's quite common. And as you know, they have a mild mental retardation, they have a stunted growth, and generally, though, they don't have a long life because they have a congenital heart disease and they are very prone to leukemia. So they generally die relatively young. Other syndromes are much worse. For example, the Edwards syndrome, which is a trisomy in the chromosome 18, generally is associated to in utero death. The live birth die in 5-15 days. It's very, very little. The Patau syndrome, which is very rare, 1 in 10,000, is a trisomy again, a numerical aberration, trisomy in 13, is polydactyl, has mental and motor deficit, and the lifespan is also very short, less than 1 year. The Turner syndrome is less fatal, let's say, but also has a lot of problems: lymphedema, short stature, sterility, hypothyroidism. It's a 45X, so it's 1 in 4,000 live female births. The triple X is instead that's mild effects, you know, is 1 in 1,000, so it's relatively common because it's not a big problem, because in women, one of the 2 X chromosomes is inactive. They only use 1. And this is called the bar body. So it's heterochromatic. One of the 2 X chromosomes is totally heterochromatic. In the triple X syndrome, both are heterochromatic. So generally, there are no phenotypic effects. But in other cases, instead, you have a lot of phenotypic effects, like in the Klinefelter syndrome, which is very frequent, 1 in 800 live male births. It's a 47XXY, so it's a male, but it's a double X. And in this case, you have gynecomastia, hypogonadism, tall stature, and osteoporosis. Much worse, but fortunately much more rare, is the cri-du-chat syndrome. Cri-du-chat is French, means a cry of the cat, because of the facial expression that is very peculiar, let's say. It's not a numerical aberration, it's a structural aberration. It's the deletion in chromosome 5 in the band P15.3. Severe cognitive, speech, and motor delays, and these unusual face features that looks like a cat. The Wolff-Hirschhorn syndrome, very rare, 10 to minus 5, is a deletion in 4P, but very serious problems. There is microcephaly, growth and mental retardation, heart and congenital heart disease. The Prader-Willi syndrome is 1 in 50,000, deletion in chromosome 15. It's a prominent nasal bridge, soft skin. You have is one of those disease where, in extreme cases, you can hardly touch everything. The skin becomes immediately red. Hypogonadism and obesity. And the YY syndrome, which is actually 47XYY, where you have increased growth velocity, tall stature. There was an early study that associated the double Y with the aggressive behavior that would be consistent, you know, with the male, with excessive male. But these studies have never been confirmed. It was a sort of fake, so it's not true. I mean, but so you see that the chromosomal syndrome are generally pretty serious in humans. Now, there is a correlation of chromosomal aberration, in particular asymmetrical chromosomal aberrations with cell killing. Why is that? Because asymmetrical chromosomal aberrations have two problems. One is that when you go into mitosis with the dicentric, for example, you enter in a position where the two centromere will be pulled by the cytoskeleton on the two sides of the cells. And so you form this bridge. This bridge can be broken, and then you enter in the breakage-bridge fusion cycle that we have seen that leads to genetic instability. But independently of that, you lose large acentric fragments. And this happens also if you have a ring. If you have a ring, you lose large acentric fragment. Generally, the loss of this large acentric fragment is incompatible with life. So this cell will die. You see, if you plot surviving fraction as a function of the fraction of non-aberrant cells, if this is the curve where you have a one-to-one correlation, you know, you have 10% non-aberrant cells, 10% survival. If the real curve is here, means that you have a lot of non-lethal aberrations. If the curve is here, this means that you have a lot of death, which is not related to the mitosis, to the chromosomal aberrations that you see. For example, cells go into apoptosis, so you don't see the chromosomal aberrations. Concerning late effects, cancer, the situation is much more complicated. We have seen that cancer is a very multi-step process. So for liquid cancers like leukemia, there is quite a strong correlation, like for Philadelphia chromosome, between the chromosomal aberrations and the cancer. Because in the case of leukemia, which is strongly related to the hematopoietic stem cells, if you induce a mutation, a chromosomal aberrations, you can have a cancer. Otherwise, the situation is more complicated. However, if you look at the chromosomal aberrations in the blood of normal individual, this is an old epidemiological study, actually. This is done in Scandinavia. They looked at chromosomal aberrations in the blood, in the lymphocytes, and they divided a large population taken from many labs in those with a high chromosomal aberration number, mid and low. And you see, this is the incidence of cancer as a function of the time since the test. They say the survival without cancer. You see that for those with high chromosomal aberration, you have a tendency to have a higher risk of cancer than those with low chromosomal aberration. For this reason, chromosomal aberrations are generally considered a biomarker of cancer risk. Now, this word biomarker is very important. Biomarkers are biological features that allow you to predict something. In this case, you can predict the risk of cancer. Now, I already explained you essentially how you form the chromosomal aberrations following the classical theory, the so-called breakage and reunion theory. The breakage and reunion theory was introduced by Douglas Lee in 1946 and is still widely accepted. Essentially, it's been refined. Now, what Douglas Lee called breakage and rejoining, we call induction of DNA double-strand break and misrepair during non-homologous end joining. But you can easily explain both the formation of simple type aberrations, you have two breaks, translocation or dicentrics, and also of complex type aberrations. You only have to induce more breaks and you will have more aberrations. there have been in the past many other theories. For example, the recombinational misrepair and the exchange theory. You know, in the exchange theory, essentially, you suppose that there is never a break. There are only sublesions that once they interact, they form an exchange. And the recombinational misrepair is something like homologous recombination, if you want. You make a break, and then this break is transmitted somehow to the homologous chromosome. But in reality, homologous recombination is accurate, and the exchange theory is not really supported by the fact that the break actually move. They really exist. They are really breaks. They can move in the nucleus to find another break and make a misrejoining. This can be seen nicely, this rejoining of the breaks, now the chromosome breaks, if you use PCC. If you use PCC, so premature chromosome condensation, you can look at the PCC breaks as a function of the dose, and this is a fairly linear dose-response curve. The number of PCC breaks per cell is generally smaller than the measured number of DNA double-strand break per cell, but this makes sense because the resolution of the method is lower. And this is the rejoining. Again, the rejoining is not totally clear that you have a fast and a slow rejoining, but some data seems to show the same thing. In addition, you can now see the kinetics of formation of radiation-induced chromosomal aberrations. You see, if you take this plot here, now this is, you see, this is the rejoining of the breaks, something similar to what you have seen in DNA repair, but this is something you have not seen. This is the formation of the exchanges as a function of the time. And if you take this curve, this is a fit essentially to this experimental point, and you overlap this curve to this one, it's essentially symmetrical. You know, while they disappear, you have the formation of the exchanges. But they are formed in different times. You see, these down here are the complex type exchanges, and they are slower in the formation. And these down up here, these are the incomplete exchanges. Incomplete exchanges, first you see many, but then they go down because, of course, you see an increase in reciprocal exchanges. So the reciprocal exchange is not formed simultaneously. First you rejoin one break, and then the other two are rejoined again. Now, how is the dose response curve? Now, we already saw this when we talked about microdosimetry, that eventually everything can go reduced, everything can be reduced to the linear quadratic theory. And this is very clear for the formation of chromosomal aberration, very clear. You see it very clearly here. So to form a dicentric, you have two possibilities. Either you have the same electron or the same ion making two breaks in two different chromosomes, and this is proportional to the dose. Or you have two different electrons making two breaks into different chromosomes, and this is proportional to the dose times the dose. So at the end, the number of dicentrics per cells will be proportional to the dose plus beta d square, and y0 is the background, let's say the background value of the chromosomal aberrations. These are now typical values for the dose response curves for high dose rate, low dose rate, and neutrons. And you see, they are very different. You see the data at low dose rate, there is almost no curvature as compared to the high dose rate. And we will try now to understand why. And the other thing is that this large curvature that you see in dicentrics is not confirmed by MFISH. By MFISH, you see that many of those that you think are dicentrics are mostly complex exchanges, and most of the dicentrics are instead formed in linear, so by intra-track events, not inter-track events. In the next lecture, we will talk about the impact of heavy ions. I want to conclude, though, going just a second back to the low dose rate data, because this is important. So if you look at the model here, or the classical model, this is the Douglas-Lee model, why the curve becomes almost linear, you see, at low dose rate as compared to high dose rate? Well, the reason is simple, no? If you have a linear component, the linear component, which will be the initial slope, you see, this is alpha, and then beta is the curvature, is independent of the dose rate. Depends on the dose, of course, on the number of electrons, but not on the dose rate. But if you want to have a quadratic component, the time of repair of the single chromosome must be much, let's say, if this is quadratic, must be longer than the time of delivering of the dose. Because, you know, if the dose is delivered at a very low dose rate, this second break will occur when this first break has been already repaired. So you will not have any exchange. So imagine that the dose rate is delivered, the same dose is given in many, many hours. The first break occurs immediately in this chromosome, is repaired, then comes the second break. But if you give the same dose in 1 minute, then you create these two almost simultaneously, and you can have a disrepair. So that's why you have a curvature for high dose rate, and you lose it when you go to low dose rate. Next time, I will show you the heavy ions.